Hello, welcome back to the Philip J. Currie Dinosaur Museum Virtual Speaker Series, where we have paleontology guests call in for a presentation on science, fossils, and dinosaurs, and where the public will be able to ask questions live on YouTube. First off, we want to acknowledge that we are on the homeland of the many diverse First Nations and Métis people whose ancestors have walked this land since time immemorial. We are grateful to work, live, and learn on the traditional territory of Treaty 8. Today, Alexis Bazinet will be presenting bird and feather fossils from the Maccabee fossil beds. In April 2020, Alexis graduated from the University of Victoria with a Bachelor of Science honors degree majoring in biology. In the last few months, she has completed a research project as part of an NSERC guided by her supervisor, Dr. Victoria Arbour at the Royal BC Museum. Alexis is excited to share all she has learned about paleontology with you. Thank you for joining us today, Alexis. I'll let you take it away.
Hello there. Thanks for that introduction, Cameron, and thank you for having me at the Philip J. Currie Dinosaur Museum for the virtual speaker series today. I'm coming to you from Prince George, BC, and I'll be telling you about bird and fossil feathers from the Maccabee fossil beds today. So the Maccabee fossil beds are located approximately 13 kilometers from Cache Creek off Highway 97, which you can see is the star here on the map. And the fossils at the site are about 52 million years old, and the site itself is about 500 to 2,000 meters above sea level. It would have been a temperate climate with resembling the coniferous and deciduous forests we see on the west coast of North America today. The Maccabee fossil beds have actually been designated a provincial heritage site now for recreation, education, and research, luckily for us, and to help protect these wonderful fossils. So there are actually about 18,000 fossils that were donated to the Royal BC Museum through the Leahy-Langvin collection. And there are many plants, insects, fish, birds, and yes, even feathers in the collection. Maccabee is known for its high preservation quality, abundance, and diversity of fossils. And it is a part of a system of ancient lakes that runs from British Columbia down into the United States towards Washington. So, like I mentioned, the Royal BC Museum received this large donation from the John Leahy and David Langvin families in 2017. Staff from the museum transported the fossils and packed them up, where volunteers unpacked them, sorted, identified, painted, cataloged, and placed them into these lined drawers you can see in the photos. And the last fossils from the collection were just unpacked February 14th of 2020. So almost a year to date now. And just a thank you to all the volunteers and everybody who made kind of this future research possible. So feathers rarely fossilize due to their delicate nature. And the Royal BC Museum has about 150 in this collection, which is a pretty large sample size for fossils, considering there was only 2 partial skeletons recovered from Maccabee bird skeletons. So our goal, we hope, by measuring and studying these fossil feathers to provide more information about the biodiversity of ancient birds present at Maccabee during the Eocene, approximately 52 million years ago. So as you probably know, feathers have function in flight, display, insulation, weatherproofing, and camouflage. So we have some examples here on the left. And depending on the body region the feather comes from left to right, we can have different feather types. So we can have wing or flight feathers. So these tend to be asymmetric with this shorter leading edge that prevents twisting in the air during flight. We can also have downy feathers, which have this loose structure, are shorter and help to trap body heat. We can have tail feathers, so they're more symmetrical and they have this interlocking fan shape that helps for steerings during flight, but they can also be ornamental, like in the peacock example on the last slide. We can have contour feathers. So these are the main type of feather found on a bird's body. They're fairly streamlined and they have sort of a waterproof tip and fluffy base. And depending on the species, the fluffy base can be quite small. We can also have semi-plumes. So these tend to be fluffy. They're for insulation and they're usually hidden under the body feathers. We can have phyloplumes, so these sense the position of the contour or the body feathers. And lastly, we can have bristles, so these help protect the bird's face and eyes. So I think in the collection, we've seen almost every feather type except bristles and phyloplumes were the only ones that we did not find. So here you can see the different feather types. So the tail or contour feather type is the most common in the collection, but we can also have flight and downy feathers. And we can also have differences in anatomy between feathers. So we can have pennaceous feathers and pumulaceous feathers. So just to go over feather anatomy really quick, the basics. Every feather has a central rachis, which is this quill-like structure running centrally. And then off the rachis, we can have these soft branching barbs, and off the barbs, we have even more branching barbules. And you can see in the pennaceous feathers, the barbules, they interlock to form these stiff, flat feathers with their hooks that interlock to form this wind and waterproof barrier. So the rhyme I learned was to fly and stay dry. The other feather type is pumulaceous, and the barbules tend to be flexible and long, and they help to trap air close to the bird's body for insulation. So modern microscope techniques can be used to investigate the fine detail of the barbules. So mostly these nodes are these kind of nubs along the barbule have different spacing, and the hooks that help to interlock between the barbules are associated with different species. And we're a little bit limited by our preservation quality. So I can see the barbules in some of our feathers, but because of the 2D sort of structure, the flattening in the slab, it does make these smaller scale features like nodes and hooklets harder to see. But these modern techniques have uses in forensics, aircraft collisions, food contamination cases, as anthropological artifacts, and in prey remains analysis. Just to give you a little bit of background about feather evolution. So we think a simple tuft becomes a complex interlocking structure through gradual stages. So we think the earliest feather was just a simple hollow tube, and then it evolved into a cluster of barbs. And based on the barbs, the base of the barbs fused to form this central rachis, so that quill we were talking about before, and the barbules branched from the barb. So we can kind of think of that downy feather again. And the barbules in number 4 evolved hooks to form interlocking flat veins. So we can kind of think of, where's my key? Contour feather, you can see the interlocking in the illustration. And the last structure in feathers we think evolved was asymmetry. So this helps with the aerodynamic properties of flight feathers. And I had to throw in a dinosaur here since we're at the Philip J. Currie Dinosaur Museum. So what's interesting about this is they recovered a skeleton from a theropod dinosaur, or ornithomimosaur, hopefully I said that right, which is the bird mimic dinosaur, which previously we didn't know had feathers. So it possessed, I'll just go back, stage 3 and 4. So it's kind of this stage and this one that we talked about, wing-like feathers, and stage 1, hollow tube feathers over the entire body. So on the right, we have an illustration of what we think an adult would have looked like. And this suggests the origin of plumage and avian wings. So we know birds are distant ancestors of dinosaurs, and we think these feathers weren't for flight, but they function in display, insulation, and camouflage long before that. Besides the type of feathers that we have that are preserved in shale slabs, they can also be found in amber. So this always kind of reminds me of Jurassic Park, but the feathers preserved in amber capture structural detail in finer detail down to the micrometer scale because of the 3D structure. So we're able to look at the pigmentation and structure of the barbules, and this tells us a bit more about the evolution and diversity of feathers. And just as an example, I think on the right, they're feathers of a non-avian dinosaur. And they figured this from the primitive structure of the barbules. And it's actually from Alberta, Canada, the one on the right. Going back to Maccabee, so we have 2 partial skeletons that were recovered from Maccabee. A skeleton of an unidentified bird, and that photo always kind of looks like a dancing chicken to me or something silly. And then we have on the right, we have evidence of a diving bird, a gaviform bird, which is pretty rare. And all represent bird fauna previously unknown in North America during the Eocene, so 52 million years ago. And if you're like me, I kind of like to put a face to a name. So wings were recovered from Maccabee, referred to gaviformes, so that would be a loon-like bird. So we have a modern loon in the top left photo. Postcranial skeleton was recovered from Driftwood Canyon and assigned to Songs a Day, which is a rail, so the top right. So 2 sort of waiting birds. And then we have 2 skeletons, one from Driftwood Canyon and one from Maccabee, referred to coliformes, which is the mouse bird on the bottom right photo. An interesting thing I learned about the mouse bird is there were 17 species widespread in North America, Europe, and Africa during the Eocene, but there's only 6 species found alive in Africa today. So the range has shrunk quite a bit. And a zygodactylid. So this is the successor taxon to the crown passerine or songbirds. So we have examples of modern songbirds in the bottom left photo. And these were found in North America and Europe during the Eocene. So this is, on the right, we have a photo from a skeleton of a zygodactylid recovered from Maccabee. And then in the red, we've circled the locations of the feathers in a halo-like fashion around the bird. We can see, so I've kind of blown up one of the feathers. So image 009 is found right here in this red circle. One interesting to note thing the authors noted was this sulfurous coloration, so this yellow, on the skeleton, which referred to the metabolic activity of sulfide oxidizing bacteria. So this kind of refers to the anoxic conditions or the oxygen-free, no oxygen at the bottom of the lake where this would have been preserved. And I'm just going to warn you that my next slide is a bit gross. It has a decomposing bird on it. So I guess you can think, why am I talking about decomposition? It has to do with us learning more about the fossilization process and how these fossils got preserved and came to be. So this was kind of a funny study where they took bird carcasses and put them at the bottom of fresh water bodies to see what would happen to them. So the process goes something like this. So the carcass remains intact for about 1 to 3 days as it floats on the top. and soft tissues start to break down by bacteria. So that's sort of A and B in the photo there. And then the skeleton remains complete, but loosely attached items like feathers start to degrade, and the skeleton starts to disarticulate. So it starts to fall apart due to scavenging, so fish or other things from the top picking at it. So we have, I guess, C and D, the skull has detached, and then E and F, the legs, which are sort of funny in that photo. And depending on the environment, so the current, scavenging, the burial, the skeleton can actually remain mostly in position in the sediment. Just to show you here, so we have muscle of an osprey or a bird after 1 day of decay on the left under a electron microscope. And then on the right, we have an even more zoomed in photo, and we can see the bacteria have already started to colonize the muscle of the bird after just 1 day. So there are these pill-like structures, the bacteria. And what I learned that I didn't know before was there's bacteria present in the gastric, circulatory, and respiratory systems of birds that colonize from within and from wounds in the surrounding and through open wounds from the environment upon death. And that's sort of what we're seeing here. And one interesting fact they noted from this study that was relevant to our own research was that there was a bias towards inland water habitats in the fossil record. So you're much more likely to find wading in terrestrial birds. True aquatic birds are absent. And we think this is because aquatic bird diversity tends to go down with the depth of a water body. They prefer to actually forage in shallower water. So maybe this is a reason. And we know Maccabee was this steep, deep lake with an anoxic bottom. So also the feathers of aquatic birds tend to have resistance to water penetration, which might not make them good candidates to fall to the bottom and get preserved. And here we can just see some examples. This is a figure I took from our paper that's in review of just different feather types and scales and the differences in preservation quality. So they're not all perfect and beautiful, but some can be measured and some can't. But just to give you an idea. So the majority of my project was actually done remotely here from Prince George. During the pandemic, we had to adopt kind of a flexible research project. And sort of how it worked is my setup's on the left, and Victoria Arbour, she's the curator of paleontology at the Royal BC Museum and was my supervisor through this project from July to October. Her setup's on the right. And she would scan high resolution microscope images of the fossils. So you can see the microscope in that image and upload them on the computer. And you can just see here, I'll give you an example of some of the fossils in the tray on the left, and then on the right, you can see the difference in scale between the resolution. I can see the barbs on the computer versus how tiny some of the feathers actually are in real life. So you can see that feather, for example, is smaller than Victoria's thumb. But I would take these uploaded photos and I would measure the feather structure. So the barb spacing, distance between the barbs is 2D. And then we have the barb diameter, 2R. And we measure barb density and total lengths also. And differences in waterproofing and insulation between bird families can correlate to different values in these properties and porosity in the feathers, and help us correlate between behavior and different habitats these birds might fall under. So we compared our measurements to modern bird families to ID any associations between habitat and species. So here's an example. So the same feather, but from different views. So one that's totally zoomed out, and then this is one of, say, a barb and all the barbules coming off. And then on the right, we have an example of how I would have measured it. So You can see you pick an area, maybe a couple millimeters of the feather, and you can count the number of barbs to get the density, and you can see all my little lines where I took the spacing and the diameter. And then we came up, and then we compared against some other papers who had published data for modern. So we compared our data to data for modern birds. And so I have some box plots here, which is basically a summary of all our measurements. So the line is an average, and then the box is the spread of the many, many measurements of the feathers that we took, and we can identify patterns from this. So we can see the habitat on the x-axis, the pennaceous barb density on the y, total feather length on the y, and we can see Maccabee here is different. It differs then compared to the other modern bird families that would be grouped by habitat. And we can also see this in the figure on the left. So we have barb spacing on the y and barb diameter on the x. So this kind of gives us an idea of the porosity of the feather. And we can see in the black, all these numbers are the different feather IDs from Maccabee. And here we have the different modern bird families, and we can see it's, clearly there's something different here. So what we ended up finding was about 75% of the Maccabee fossil feathers in our data set were flight and contour feathers. Barb measurements varied slightly by measurement region, so whether you measure from the bottom, the middle, or the top of the feather. So for our figures, for example, we just chose to look at the mid-vein region of the feather. And the Maccabee fossil feathers showed unique substructural properties not present in modern birds. So they were smaller in size, they had a higher barb density and low barb spacing. And the Maccabee fossil feathers are likely not derived from aquatic birds as predicted. So originally we thought, okay, it's an ancient lake, we're probably gonna have feathers of some aquatic water birds in here, and that's just not what we found. So we think the Maccabee fossil feathers are most likely from small arboreal birds or wetland birds inhabiting the forests surrounding the lakes. So like I said before, wading and perching birds. If you'd like to learn a bit more, you can always check out the Royal BC Museum Learning Portal. There's some other photos if you want to see like the insects and the leaves and other things in the collection. And we recently submitted our research as a paper to the Journal of Vertebrate Paleontology, and it's currently in review. And though we could not identify specific families of birds at Maccabee, it does broaden the amount of vertebrate fossils from the site and the bird biodiversity. So you can look for that title. All right. Thank you.
Wonderful. So now we have a time for our question period. And here's our first one. When should we expect to see the publication?
So it's currently in review, but I'm hoping this year.
It's hard to tell.
Yeah, hopefully in the next few months we'll have an answer.
You bet. Is there any other paleontology research projects you'd like to be a part of?
So I actually did my degree in marine science. So I would be interested maybe in a project looking at marine mammal fossils. I saw, was it somebody from the UK? I attended a conference and she scanned all these whale skeletons and was trying to learn about their evolution. And yeah, it was really interesting.
That is very cool. Do you know if there's any more plans for the Maccabee fossil beds?
So trying to think. So Maccabee is actually probably the most famous for its diversity of leaf, plant, and insect fossils, which is also how we know about the climate and sort of environment that would have been around that site at the time. But I think there will be more that will come out of it because we're just starting to study the feathers, for example. And I know pre-pandemic, my supervisor, Victoria, was excited to do some field work trying to look for other fossils in northern BC. So I think there's a lot more untapped research in paleontology in BC to come.
Exciting opportunities. Richard Doucette would like to know, you said that the Maccabee site would have been a deep lake. How deep would the water have to be to have the lack of oxygen to preserve birds?
I don't know if I have a specific depth for you. We just know based on the sulfurous coloration of some of the bird skeletons and based on some of the geology of what the site looks like today, it's sort of a steep slope. And the fossils are preserved in like these shale slabs. So I think if you actually, I haven't been, I'd like to go, but if you've been to Maccabee, you would see like the dry cliffs and then all these stacking layers of deposited fossils and soils that got like squished over time. Yeah. But yeah, I'm sorry, I don't have an exact answer for you. I'm just thinking back to marine bio background of Saanich Inlet in Victoria, and that is also anoxic bottom of the ocean there.
True. Well, Richard would also like to know, could the challenge in comparing to current birds be because of flattening and compression?
Yeah, so I guess there's three things I would say. So one of the possibilities is that some of the differences we're seeing in the feathers could be from taphonomic changes, so from the fossilization process. One could be because there's a lack of data, a lack of modern birds and extinct birds to compare to in the literature. So we only had 2 studies that we could find that had a really good array of some of these measurements for modern birds. And then 3, it could be a real biological signal. The reason, I don't know, Victoria and I don't 100% know if it's a taphonomic bias. So you think if you squish the feather, the barbs would actually be thicker or wider, but that's not actually what we found. We found smaller, they're smaller barbules than, anyway, you think if they'd be flattened, they would be larger, not smaller, but it's just not the case. So we're hoping it's a biological signal.
That would prove quite significant. And Richard Doucette would also like to know any new cool finds found during the unboxing.
During the unboxing. Well, I haven't got to, so originally I was supposed to spend my summer research just helping in collections, boxing and cataloging, and maybe doing a research project. And then with the pandemic, everything went remote. And so we did a full-on remote research project instead. So I actually haven't got to volunteer behind the scenes with the unboxing. But yeah, I can just tell you that I was personally surprised about just all the different feather types and the differences in preservation quality. And I know like surprises, I guess what's rare about Maccabee, they have insects. They found crayfish in the collection, which I think is pretty rare, and a few other things like that. So
cool. Yeah, it's hard to preserve feathers in the fossil record. There's not as many as you would think. It's a pretty
Yeah.
special site.
Yeah, like the only two we can kind of compare to, but they haven't done a study like this yet, is from the Eocene, is the Messel site in Germany and the Green River Formation in the United States. So Victoria and I are kind of hoping the Green River Formation is really extensive, that, if other museums kind of pick up on doing these studies with isolated feathers, maybe we can get a refined method going and learn a little bit more about differences in the environment between sites. And anyway, I'm going on and on, but
That would be fantastic. There's a couple more. Who is on the paper that is in review?
So it is myself and my supervisor, Dr. Victoria Arbour from the Royal BC Museum.
Awesome.
So Victoria actually does her research in ankylosaurs, so those spiky dinosaurs with the club tails. So it's both our first adventure into the Eocene and feathers and birds, the ancestors of dinosaurs.
Very cool. A question about the software that you use to virtually measure these feathers. What did you use?
Oh, yeah, I forgot to mention that. We used ImageJ, so I believe it is a free software, but you can upload the photos as a TIFF file, I think is what we did, or a JPEG. And then, yeah, you can they have a line tool, and you can just measure. If you have a scale, you can set the scale and then measure right on top of the photos.
Very cool. And I think you already answered this one, but I'll still ask it. In the future, do you want to visit the Maccabee fossil beds in person?
I think, I've done the drive between Prince George and Victoria and Vancouver so many times, and I never knew it was there, and I would really like to go. Yeah. Maybe when it's nice and not winter for all of us, but summer maybe.
Exactly. Well, I don't think I see any more questions in the comment box. I think that'll do it. Thank you so much, Alexis. Join us again Saturday, February 20th at 3 p.m. as Michael Hudgens will be giving a talk on the paleobiology and evolution of Thescelosaurids, a dinosaur that has recently been found in the Grand Prairie area. Take care.
Okay, thanks everyone.